So we know that today the main cause of death and the main component of the healthcare cost is the so-called non-communicable diseases, which have overturned infectious diseases both in developed and in developing countries. And the WHO goal is a 25% reduction in these diseases by 2025. And most of the interventions which are carried out today is about adult interventions. So series of randomized controlled trials are run both in developed and developing countries about reduction in unhealthy behaviors in adults. Okay, so try to eat less salty, try to exercise more, try to smoke less. And the extent to which these interventions are actually working is still, under scrutiny because they're very short follow-up. But what I'm going to show you now is how something happening early in life can have long-term effects, both detrimentally in cases of bad early life conditions, but long-term positive health effect in case of early life enrichment. And just to get back to what Professor Ekman was showing you before, if we just look at disparities by one measure of socioeconomic status, which is education, not surprisingly, what we have here, the more educated individuals earn more, are more likely to be employed, are more likely to be better off on a variety of different dimensions. And if we try to do a simple decomposition, like we did in our work, between the average treatment effect of education and then the selection on early life factors, what we do see, I think what's interesting is that especially in terms of health stocks, so depression, self-reported health, obesity, a big component, at least half of these disparities due to factors occurring in the first 10 years of life. And you see that the education has a bigger effect on healthy behaviors rather than health per se. And again, in this paper, we further decomposed the selection component into selection on traits and selection on parental background characteristics, and we found that the bigger component is due to selection on early life traits. And this simple version here, we had only 3 traits. We had cognition, self-regulation, and early life health. And then we had other work where we have expanded the set of allowable traits to reach 13. And so, as I said, what I want to focus on is basically these driven factors, attachment, stimulation, and nutrition, and emphasize what are the mechanisms through which they might affect late life health. And in particular, I want to focus on 2 sets of mechanisms, biological mechanisms and socioeconomic mechanisms. Now, why are these 2 important? Because if you think about something which affects your health, it has to work through your body, right? It has to come from the external environment, permeates within the body, permeates within the cell. I'm going to show you it changes the biology of the body, and then it gets reflected again in your external behavior, in your phenotype. What's the point here is that in humans it's really hard to disentangle these 2 components. Why? Because whatever affects you is also going to determine not only biological but also behavioral responses. So parents can react to something, to a shock affecting the child, and then the effect that you see, say when the child is adult, is going to be a composite effect of both the biological response and investment of the parent. And it's really hard to disentangle the 2. But in humans and in the monkey work that you've done, that's very easy to do because that's a very controlled experiment. And so using animal models, you can actually disentangle the 2 mechanisms and you can understand the extent to which early life factors affects your health purely through the biology of the body. And then assuming that the biological mechanisms are the same between animals and humans, and that's something that can be investigated, the genes which work in the same way, then you can try to piece out the biological component of humans and try to understand the extent to which responses actually do matter and can amplify or can reduce the effect of early life conditions. And then, of course, there is all the effects of genes interacting with the environment. So what's the way of thinking about this? And this is very important because what we call environment here could be a parental investment, it could be a policy, it could be an intervention. And so when we look at, say, we do program evaluation or we look at the effects of intervention, what we usually do is something like this. Okay, we have an intervention like health insurance, how this affects health. What we can do today, because we actually have the technology and we have the data to do this, is we can try to understand how this works from the inside. Why is this important? Well, because actually now we already have some technology to intervene from the inside, to intervene at the biological level. So what is this showing here? I'm not sure if it's better if you see it in white or in red, and this is what it was asked before about this whole thing about gene expression, right? So what we have here, this is like biology 101, I'm not a biologist, but I'll try to tell you something about this. So this is a cell. What is in the nucleus of the cell? You have genes. What is the gene? The gene is just a strand of DNA, okay, which is the code which determines the way that we function. Now, in the DNA, you see that you have two regions. This region is called the promoter region, and this is the coding region. What happens in the promoter region? You have a stimulus from the external environment, which tells to your body that the information which is encoded in the DNA has to be translated to produce proteins and then to determine all the functionings of the body. Now, gene expression is very important because it's the way our body works, the way the genetic information encoded in the DNA gets first transcribed into RNA to make it operational and then translated into the formation of proteins. And proteins determine all what we do, determines how our body works. So that's why gene expression is really, really important. And this thing here, this is a called transcription factor, which is just the factor which binds to this area of the DNA and tells, well, start transforming into RNA, producing proteins, because I need this bodily function to be performed. So everything which disrupts the way genes express themselves can determine a change in the way our body functions towards the optimal level. And everything which comes from the external environment can actually affect the way our body works. And nowadays, we don't really know whether this is going to be permanent or not, but you can understand that it's something very important to understand how environment's intervention, parental investment even, can affect the health and then the rest of the development of the child and the adult. So the first mechanism that I'm going to look at is basically attachment. So why attachment is very, very important, and as Professor Conti said before, with attachment, just a sense of a certain relationship which exists, especially between the mother and the child, to which the mother gives a sense of security to the child and allows him to explore the world, but then to come back to have a secure basis. And there's been lots of evidence showing that this attachment mechanism is very important. Evidence from rats and evidence from monkeys. And the idea is that when you have the mother close to you, you feel more secure, you're able to explore the world. And the mechanism through which this works is through a better regulation of the stress response system. Okay, so just this stabilizes the stress response mechanism of the child so that when the child becomes adult, is better able to handle stress situations. And it has been shown that this works through epigenetic changes in a way that can be reversed. So I can disrupt this in childhood. It has been shown in rats. I change the gene expression. I become more susceptible to a stressful environment, but through this administration of epigenetic drugs or through environmental enrichment, this can be reversed back. Okay, I can be still better able to handle the stress. This has been shown in animals, not in humans yet. Okay, what has been shown in humans, there is seminal work by Bowlby in 1951, which has shown that the presence of the mother is very important to determine the absence of any mental health problems. And this is studying of children, orphans, deprived, raised in the orphanage. And there has been other work related to the presence of adverse conditions in childhood, maltreatment, and it has all been shown that this has led to very bad health conditions, especially related to inflammation and chronic conditions, which pretty much work through a disruption in the stress response system. So what I'm going to give you now, a little bit more evidence on monkeys, and probably if you remember from before, in these experiments, monkeys were randomized at birth in triggering conditions. Now if you're going to ask why monkeys, there are at least two reasons. You cannot do this experiment in humans. It's kind of unethical to take children away from the mothers, at least on purpose. Two, you can, And then the monkeys are actually very similar to humans under many respects, especially this kind of monkeys which are rhesus monkeys. So what happens here at birth, monkeys are randomized in 3 rearing conditions. One of them are left with their mother since birth, so this is like the standard maternal rearing condition. Then there is another group which is taken away from the mother at birth, and they are put in a cage with other 3 monkeys of the same age. So these are the so-called period monkeys. And then there is another group which is put in a cage alone for 22 hours in a day with a surrogate mother, which is just this hot water bottle with a covered cloth hanging from the top of the cage. And for 2 other hours a day, they can play with other surrogate period monkeys. So this is a sense to give you a normal, rich environment, to give a sense of maternal deprivation, but not social deprivation, and this is total maternal and social deprivation. Okay, now you can imagine that if you are, raised up in these conditions, you cannot end up really well. And so what we have done with this experiment so far, we've done 2 studies. In one study, we have been looking at the effect of these 3 conditions on changes in gene expression at 4 months of age to see if being reared in adverse conditions can actually change the biology of the body very early. And then in another study, we looked at after the end of this experiment, because after 1 year, all these monkeys have been put together, okay? Those reared with the mothers, those reared without, to see if there was any evidence of remediation or reversibility. And so I'm going to show you a little bit of evidence from these 2 studies. So what do we do in this first study? We look at the effect of these 3 rearing conditions on the genome-wide transcriptional profile of circulating leukocytes. What we found that there was a change in gene expression. And this is the heat map that was showing you before. So how to read this? Each of these columns is a gene whose expression has changed between mother-reared and surrogate period monkeys. Each row is a different monkey. So you see the sample size is not too big here. This was a pilot study, now we're having a big longitudinal sample. What is really interesting here is that the red part here, it means these are genes which are at least twice as much more expressed in monkeys not reared with their mothers. And the green part here, so you see, you can only see them in relative terms because the absolute magnitude here doesn't really mean much. This is our RNA. Yeah.
Just a quick question. Yeah. What do you mean by expression?
Expression means the amount of genes which are encoded into proteins. Right. And so here, that's why I'm telling you, you can really think of this just in terms of relative terms, because if I tell you the unit, this is just optical units, because the way this is measured is that you have on the microarrays and you see how much is fluorescent the light. The more fluorescent, the higher the level of gene expression. It's really like spectrometry. Okay? And here we only looked at genes which are at least twice as much differentially expressed, and then we did all the same thing that we did for the early childhood intervention to control for small sample size, multiple hypothesis testing, because of course you went through all the genome. And then this is really just looking at mean differences, but we also replicated the same results looking at the overrepresentation of genes in our sample in these differentially expressed genes versus the full genome, and those were confirmed. So we really did everything we could. But what is really interesting is that if you look at what happened to the monkey not reared with their mothers, well, their genes are much higher expressed, the ones which determine inflammation and control cell growth and differentiation. So it means, basically, if you're not raised with your mom, you're much more likely to have an inflammatory response. Why? Well, because you're stressed, you're alone all the time, and there is nobody there for you. Okay, and if you look at the genes which are underexpressed, so they work, let's say, less optimally than they should, these are genes related to antiviral response, to interferon, which means that if you have been attacked by a virus, you will be less effective to respond, you will be more likely to get sick. This is pretty much consistent with the human evidence that we see in children reared in adverse environment, exactly the same time of inflammatory response that we would see in adulthood, the same type of lack of defense to viruses.
Is there any evolutionary reason why this would be an optimal response to the poor environment?
Yeah, there is actually an evolutionary reason. And the reason is that if you think about the way in which pathogens occur in your body, no? So our viruses transmitted versus our bacteria transmitted.
I was wondering if like one maybe limitation of taking it, applying this in humans is that children might have a chance to actually seek out other paternal figures in their lives.
Yeah, that's a very good observation. Let me give you the answer to this one. So that's exactly one of the things which are doing now. Because, okay, so the way this experiment was set up was to say, okay, let's see if you actually can remediate just by putting together children born in adverse and normal conditions. And the evidence that I'm going to give you after this one, the one after the end of the experiment, shows that there is no remediation. What they are doing now, after we've shown these results, they've actually changed the experimental protocol, and now there is, in addition to having the children all together, they put what they call the foster grandparents, which is slightly older monkeys, and it shows that there is some evidence of remediation in that case. So that's the usual critique that we get at the beginning. Yeah, but you can put the children in foster care. These were not in foster care. But now if you put them in foster care, it actually looks like it's better. I don't have those data yet, but this is what's being done now in the lab, in the primate facility.
Gabriella, could I just clarify? I thought that Sumi had some studies, not grandparents, but just putting these monkeys back into normal environments.
That's what we do here, but that doesn't work too well. It works well, so you have a remediation of the behavioral response.
Right, there was some modification of it.
Yeah, that you have. That's actually in the other set of slides. But for health, you don't have a remediation.
No, not for health, but for some of the ventilation.
That's true, yeah.
So there could be some other things.
Yes, exactly. You have a remediation for behavioral response, and then for methylation, that you have it for the women, the female monkeys.
The female monkeys?
Yeah, the female monkeys. Right, not for the males. The males are, once they're big. Anyway, so.
Why should that be, though? Why should it be?
Because females are more resilient.
You're saying biologically now. I mean,
yeah, they're biologically, adaptationally, evolutionary, whatever reason you can think of. Okay, and so we were looking through mechanisms, by which this can occur, because if you think, there is the external environment, how this relates to changes in gene expression, it has to work through the brain. The brain must send some signal. Okay, and then the second question that we asked in the second paper, which was actually the first that we wrote, was that, okay, so the monkeys now, we've seen that there's serious disruption at biological level. If we put them all together back in a normal environment, are these reversed? And as we were saying, there was evidence before that this had been, yes?
So a question on the earlier results. So, these are in a particular situation where you're putting monkeys in kind of a less social environment. And you're kind of pitching this in terms of a stress response. Have there been other studies that have looked at specific stress responses and found the same kind of results, the same kinds of genes? Or
right. So this is the first experimental study.
So no other studies
that exist. You have in monkeys, at least, or whatever it was in humans. Similar, there have been studies in rats which have shown similar patterns. Those were to prenatal stress. So this is postnatal stress. There is evidence in humans, and actually our coder has done some studies in humans, which was the one on social isolation he was referring to before. And the mechanisms and the changes that you find are very similar. The problem with the human studies is that they are observational, and so that's only correlational evidence, and the sample size is it's bigger than this one, but to be an observational study is not that great. So, but yes, I would say that the evidence is consistent from what I know and I've seen so far. Yeah. Okay, and then what we're asking is whether there is any evidence of reversibility here, and what's the point is that if there is any evidence for behavioral mechanisms, there is, or at least we don't find any evidence of remediation in terms of effects on health. And so what we find is that we looked at the effect on physical mental health, and we found that the effects of this early recognition were still very much visible and evidence both in terms of physical health and mental health. Yeah.
So presumably these monkeys are
socially not the same as monkeys that were reared by their mothers.
Yeah.
So throwing them back into a cage with monkeys that have normal rearing.
Yeah.
What's the dynamic, and is that relevant?
Yeah, that's a very good observation. So if you look at them in this big cage, what you see is that the monkeys were raised with their peers. They just cling to each other because they cling with the peers they were raised with, and the ones who were raised in isolation, they are just still, they're on their own. So it's actually this normal environment is not doing, much. That's why it's important that when you put like the foster grandparents, because they go to them and they try, to change and to give basically that attachment which was missing when they were younger. This is why, another figure is more important. And you can think, that you call it a normal environment, that it's not really their normal environment. If you're raised in a certain environment, you might just be used to it, and you might be better off under stressful condition. Actually, there is evidence that if you're raised in very stressful condition, you're much better reacting under stress, because that's like, that's my normal environment. It's I was raised. And so we were actually really lucky with this one, because what we found is that these monkeys, by standards of the animal committee, they had quarterly veterinary visits, and so we had very, very good detailed health records. We prescribed all of them, we put them in digital format, and we were able to analyze the health effects. And so what we find, these are the monkeys raised with the surrogate. And what we see is that they might have much higher probability of developing illness. And notice the effect of physical health are only present for the males. There is no effects on physical health for females. Which type of physical illness they were more likely to develop? Those again related to stress conditions. In particular, they were really like stress related, and we found evidence of changes in the blood tests that we had. And then we looked also at results on mental health, and this comes from observation performed by a skilled technician who was observing the monkeys at random times for a 5-minute observation window, and they were looking for whether these monkeys exhibit behaviors which we would call something like, autism, ADHD types of behaviors in children. And then, not particularly surprising, monkeys not reared with their mothers had much higher probability of developing this form of abnormal stereotypical behavior, both for males and for females. And this is after 1 year, so this is between 1 and 4 years after the end of the experimental period. Still, you see this effect of early life condition long lasting, and no sign of any remediation in this case. And so also in this case, we wanted to understand which are the mechanisms through which this early life condition translates into late life outcomes. And what we had for these particular studies, we had very good measures of stress-related hormones like cortisol, HCTH. And these were taken within the experimental period. And so what we found is that if we look at the male monkeys, what we have here we have the ones raised with the surrogate, and here are the ones raised with their mothers, and these are the ones which developed physical illness, and these are the ones which had lower probability, and you see the level of cortisol in monkeys raised without their mothers is much higher. And what do I have here on the left? I think this is really interesting. I told these effects in the monkeys are very much similar to what you see in humans, and so if we look at this left part of the graph, what we have here, this is the level of cortisol which has been shown in children raised with their biological parents and children raised in orphanages. So it's exactly the same types of biological response, and it's exactly the same type of health outcome.
You mean on the left-hand side, human?
Yeah, that's humans. Cortisol in humans,
So
but not our side.
When you say biological parents, do you use grandmother?
It was the parents, like the biological parents. This is like a study by Gabriella and others, and these were.
What about the grandparents?
I don't know about the
So
grandparents.
the orphan are not. That doesn't include the grandparents either.
No, this is just the monkeys raised with the mothers, with the surrogate.
The orphans one, the red one. Would you say orphan?
For the humans.
The humans are in an orphanage, no grandparents.
So you don't, there's nothing in between, right? Because in developing countries, you see a
No,
lot of.
it's just because justice.
I mean, most of the kids are not going to be orphans. Most of the kids will be with grandparents.
No, but this is also okay. This is children with biological parents in U.S. and this is orphans raised in the orphanages in the Eastern European countries, where then came into U.S. And as soon as they came and they were allocated to families in the US, they measured their cortisol, and what they found is that the children were with their biological parents at much lower levels as compared to children who were brought into families in the US, but until then were living in orphanages into former Eastern Europe. Okay, so no grandparents anywhere here. Okay.
Do you have the orphans once they're adopted out though?
No, I don't have
Yeah,
that.
that's
I mean,
just-
I guess it exists, but I don't
So
know
these are
here.
Romanian orphans.
I can't remember whether this one in particular is a Romanian.
Right.
Yeah, but it's that subtle study that Megan Lunar is done with all the cortisol measures. Yeah. Okay. And so, is there anything we can do about this? Okay. So, if attachment is really important, lack of attachment is these adverse effects. It works through biological mechanisms. Is there anything that we can do in terms of early life interventions? It actually looks like there is a pretty extensive literature, which is summarized in this meta-analysis, on the effectiveness of early attachment interventions, and in particular, some of them which are particularly effective seem to be home visiting programs, such like the nurse-family partnership, which works on mothers in the prenatal period, pregnant mothers, nurses visiting them at home. Now, what is even more interesting, though, is that, okay, early life intervention works. What about the possibility of intervening later in life? Okay, and there is evidence which is accumulating in the recent years that some of the negative effects of this early life experience can actually be remediated at a later period. Now, so far, the evidence mostly comes from animals. There is some evidence on humans, actually, in the stimulation section I'm going to talk about now. And how does this work? So it has been shown in rats that rats which were deprived of their mother, and so they were raised in a similar situation as monkeys, in the adolescent period, so in the puberty years, when they were given environmental enrichment or metal supplementation on histone deacetylase inhibitor, which are basically epigenetic drugs, okay, are ways of improving the gene expression, which was lowered by early environmental disruption, the effects of low maternal care on stress reactivity can be reversed. Okay, you just are as good as if you were with your mother at handle stress, both in terms of behavior responses. Sometimes there is also reversibility of the gene expression level. So that's all very interesting. I don't have any evidence on this apart from rats. Okay, no evidence on humans yet. So this was the first factor that I want to talk about.
But the orphan studies, the studies by Rudder and Nelson and the guy from Maryland.
Yeah, but that's mostly, so I'm talking here mostly in terms of health effects.
Right, but
Right.
there do seem to be, at least in terms of the social.
Yeah, so
measures.
the social, exactly. So my understanding is
The kind of cognitive behavioral,
Yeah, yeah.
partial remediation.
exactly, yeah. So usually the cognitive, the behavioral component is kind of the easier to remedy.
And they haven't taken health measures.
Not that I know of. Not that I know of.
Easier than
The health.
than health.
Yeah, it looks like the health is kind of a bit trickier, but the behavioral responses are the ones which looks like.
But it's not full remediation. It's partly depending on how old they are.
Yeah, it depends on how old they are taken out of the orphanages and they are put into families.
Again, that's the continual notion of complementarity getting stronger and stronger with rigidity, at least in some dimensions. But the health, frankly,
Okay, so the second factor I want to talk about is stimulation, and really in terms of stimulating the brain while it develops. And why is this important? Well, there are many studies on animals which show that if you block, for example, the visual input for one eye, so if you don't stimulate the particular organs which is related to that brain function, that actually that part of the brain is less developed in terms of neuronal connectivity, dendritis, densities, and so on and so forth. If you don't stimulate the area of the brain related to that particular function, actually underdevelops. And there has been lots of work in humans on, for example, giving children exposure to second language in the first 6 months changes their phonetic perception. The study that you mentioned before about the number of words read stimulates the development of neural synapses, and as today we know pretty well at which stages the different areas of the brain related to sensing, language acquisition, higher cognition develops. So that's great. Why does it matter for health? Because you can think that stimulation of the brain forms of these traits we're talking about before, cognitive traits, other mental traits which can affect health, either through healthy behaviors or, for example, through acquiring higher education, which affects health, or through acquiring a higher income, which also affects health. And there is a lot of evidence now in economics that these traits do matter either directly or indirectly for health. And so what I want to give you now, I just want to give you some evidence on 2 of these traits. One, I'm focusing on these 2 because these are exactly the 2 traits that I'm going to show you have been affected by early childhood interventions which have an effect on health exactly in this behavior and in this talk that I'm going to show you here. So this comes again from the study that I have with Professor Ekman, where I show you the differences in health disparities before. And so what is looking at? This is looking at the effect of age of traits measured at age 10 on the probability of being a daily smoker at age 30. And we have 3 of these traits, and these have all normalized that the bottom distribution, they are normalized at 0, so we can actually compare the effects across them. So what do we have? In black we have cognition. Then in red we have traits which I'm gonna call self-regulation. It's really the capacity of self-regulate and the focus on the task and do not lose your attention. And then in blue I have what I call health, and this is really a measure of physical fitness of the child. Okay, so the child being physically fit, not being, say, obese. So what we see here, so one thing that we find in this study, which I think is really interesting, is that cognition, especially for males, actually doesn't really have much of an effect on health, on healthy behaviors. What really matters for smoking behaviors is, 1, your level of early life health, which makes sense, but what matters most is these non-cognitive traits, again, which I guess the psychologists would call conscientiousness. And you can see that if you have an intervention which is going to increase the level of the traits in this child by age 10, you will have a significant, also in magnitude, the decrease in the probability of being a daily smoker by age 30. And one thing that I want to say is that it's really important to consider more than 1 trait. We have this paper where we have really tried on the possible method to try to understand how many traits there are. Why? Yes, no.
Do you interpret this as a causal effect?
Yeah, under the assumption of the model, yes.
But there's no correction for if father or mother is a daily smoker.
We don't have whether the father and mother daily smoker, but we do correct for family and grand characteristics which are correlated with the cognitive and thyroid characteristics. Yes, I get that every time. We should get it at the time. What? We didn't. But anyway, we have exactly the same results in the paper where in the previous version where we had these 13 factors, and we always find that these non-cognitive traits actually have a big effect on smoking. And these, reflects on the fact they have ADHD, attention type problems, are likely to be a smoker. So I'm pretty convinced that that's causal, and I'm going to show evidence from the theory that if you actually prove that trait through an intervention, you're also likely to be a smoker. So that's one thing. The other thing which I think is really,
We have to go in about 4 minutes, just to let you know. We're just on 6 minutes.
That's fine. You said most of it, so I'm just gonna go through the game. And so what is really important here is that you actually have not only include only 1 trait. Why? Because this is, again, the effect of cognition. This is the fact that we saw in the model where we had all these 3 traits, so basically 0. If you have a model where you only include cognition, you will see that it has a significant effect on health, which might actually be spurious. Yeah.
On this, is it one factor that loads on everything all 3 good factors used to load on, or is it one factor that still just loads on? It's just the cognitive part of it.
Yeah. And then the other one, if we look at the probability of being obese at age 30, what really matters here is just matter how healthy you were when you were a child. You know, especially, you know, if you're not an obese child, it makes sense. You're not going to be an obese adult. And these two type of traits are exactly the two traits which have been boosted by the parent abecedarian intervention and which are driving the results that we see in the health effects later. So, since we don't have much time, let me just skip through the Peri preschool program, which you already told you before, and just let me take back again the evidence on the long-term health effects. So what do we find here in terms of the health effects of the Peri preschool? Remember, this was the intervention where the children were basically brought in for 2.5 hours a day, and they were given this plan to review task. So they had to plan the task, to do it, and to review it at the end. So you can think that this will really boost their organizational skills. We'll have to put them to stay on focus, stay on task, and then review in front of their peers. Okay? So what do we find in terms of health effects? The big health effects in the Peri intervention was actually smoking behavior. If we look at the treatment group, both at age 27 and at age 40, the treatment males were much less likely to be smokers, and if they were a smoker, they were much less likely to smoke a higher number of cigarettes. So they were either more likely to be no smoker or more likely to be light smoker. When we looked at what were the mechanisms driving these changes, what we saw, as Professor Ekman showed before, this program had a big effect on this reduction in externalizing behavior. If we decompose the effect of the treatment of probability of being a smoker by these early life traits, this gray part here, so the big chunk of the treatment effects, is actually explained by this reduction in externalizing behavior. So that's exactly, you know, these traits that I showed you before has an effect on the probability of being a smoker. We have here an intervention which reduced the probability of being a smoker in the treatment group and which worked mostly through changing this self-regulation traits, reduction in externalizing behavior. These kids had to stay still, had to stay focused, and they ended up smoking less up to age 40. Now we know what happens at 50. This was the first thing. The second thing that I show you, the effect on obesity. Okay, so let me skip through the nutrition part to the abecedarian intervention and get back to the other results that he showed before. So again, health effects of the abecedarian intervention. This was actually an intervention where the children stayed in the childcare center from 0 to 5 years, 8 hours per day. They not only had this cognitive stimulation, but they also had their breakfast, their lunch, their afternoon snack. They also had the pediatric care. So it was really a complete composite intervention. As I showed you before, what we found, big, important health effects at age 35. So the treated were much less likely to have metabolic syndrome, which is having lower abdominal obesity, having higher level of the good cholesterol, having lower level of hypertension, and so they were less likely to have each of these components. And the magnitude of these effects is very much economically significant because these are exactly the conditions which are driving the bulk of healthcare costs in the United States and in the rest of the world. And we do find that the treated group was, under every respect, much healthier under all of these conditions. And in particular, we find that the treated group has a much lower score for the Framingham Risk Index, which is an index which predicts the probability of developing cardiovascular disease in 10 years. So those are really healthy, especially the males. Now, where is this coming from? Okay, this is really interesting because it tells you something about how the characteristics of these interventions, what they did to the children, is going to then affect how they ended up. So did this come from IQ, boosting IQ, which improved their healthy behaviors? It doesn't look like. If you look at the male IQ in the abecedarian, there was fed out much later than in the Peri, but still there was. Did this come from changes in their cognitive skills, personality? Did they smoke less? Did they have healthy behaviors? It doesn't really look like in the abecedarian we had the big healthy behavior changes that we saw in the Peri. So where is this coming from? What we looked at BMI of the children in the various groups. Remember, they were eating there. They stayed there all the time. And so what we found, This is 3 months, right? So this is just after the beginning of the intervention. Now focus on this solid black line, which is the treated male group. This is the density of the body mass index of the children. Look at what happens. So this is at the beginning intervention, so not much difference. Look at what happens to the body mass index of the treated group throughout. This is 6 months. It starts shifting to the left. Look at 9 months. It's very much less spread out than the one in the control. Look at 12 months, and then 18 months, and then 24 months, 36 months, 48 months, 60 months. The children, the males who were in the treated group of the Besedarian intervention already had a lower body mass index. The entire distribution was shifted to the left. It was much less spread out than the ones in the control group. And if we look at the evolution on another perspective, the evolution of BMI over the timing of the intervention by treatment status, what you see? These are Z-scores of the BMI. So if we look at the treated group and the control, they started off pretty much the same point, while the treated group ended up pretty much the median of the reference population throughout the whole period. The control group had this surge in BMI, which continued throughout the childhood. So they were already at there of a less than optimal BMI when they were children. And if we look at how this looks like by obesity status in the mid-30s, which is when we found those differences, then the adults who were obese in their mid-30s were already on the higher than median weights in their childhood years. Those who were not obese, those belonging to treatment group, were instead already at median level in their childhood years. And this is true throughout all the time for which we have these observations in their childhood. So those health effects that we see at age 35, we can see them in the childhood years, and not through, in this case, changes in cognition, personality traits, but through changes in the BMI, in the physical health of the children. Why? Well, I guess because in this case, they were having their nutritional component at the center, and they had the free pediatric care. Yeah?
It doesn't seem to work for females.
For females, it doesn't work.
That's a reason.
We can talk about it later. That's a long story. Why there are gender differences in the monkeys, why there are gender differences here. Okay, one thing that it's easier to say is that...
So I'm sorry, I think we have a meeting across the midway in 10 minutes, so we
I'm done. This was the last one.
Thank you.
Yeah. No, I was just, yeah. The one thing about females is that if we look at the Besedarian, actually the females, the treated females were of lower weight than the controls, in particular lower birth weight. Okay, why is that? Because the mothers who were more likely to work were more likely to be the girls in childcare, and these were mothers who were more likely to work. They are usually most of them are more likely to have children of a lower birth weight. So my interpretation is that in the case of females, with intervention allow the girls to take chocolate for better nutrition to normal level. So they were not thinner, they were stronger. There was just no difference between treatment and control, but the treatment was starting to go for a suboptimal level, especially nutritional as compared to the control. So the intervention allowed them to take chocolate. So it was still a positive effect, if you like, it's just not this visible. So this is all I had to say about this, so you guys can all go through dinner, whatever you think.